Hello everyone, and welcome. I'm excited to have you here today for this presentation. We'll be exploring a comprehensive review of innovative strategies to prevent antibiotic resistance. The focus will be on the important role of postbiotics. Over the course of this talk, we'll cover key concepts, recent advancements, and potential solutions in this vital area. Thank you for being here, and let's get started. The advent of antimicrobial resistance has significantly increased the impact of infectious diseases. It's not just about more infections happening, but also about the rising healthcare costs associated with treating them. Even though we have a large arsenal of antimicrobial agents to choose from, resistance has become a major hurdle. In fact, resistance has been documented to all of these drugs, and unfortunately, it tends to develop quite quickly after a new drug is introduced. This rapid emergence of resistance is a serious concern. It's what prompted the WHO to take action, leading to the launch of their Global Action Plan on Antimicrobial Resistance back in 2015. This plan aims to address this growing threat and find sustainable solutions for the future. Antimicrobial agents can be classified into different groups based on how they actually work to fight bacteria. These groups are really important because understanding their mechanisms helps us choose the right drug for each infection. The first main group includes agents that inhibit cell wall synthesis. These drugs prevent bacteria from building or maintaining their cell wall, which is crucial for their survival. Without a proper cell wall, bacteria can't stay intact and eventually die. Next, we have agents that depolarize the cell membrane. These disrupt the normal electrical charge and stability of the bacterial membrane, which interferes with vital processes like nutrient transport and energy production. Then there are drugs that inhibit protein synthesis. Since proteins are essential for virtually every bacterial function, blocking their production effectively stops the bacteria from growing and multiplying. Another important group targets nucleic acid synthesis. These agents interfere with DNA or RNA production, preventing bacteria from replicating their genetic material and thus halting their reproduction. Finally, some antimicrobial agents inhibit specific metabolic pathways within bacteria. By blocking these pathways, the bacteria can't produce essential compounds they need to survive. Understanding these different mechanisms is key, because it guides us in selecting the most effective agent for each type of bacterial infection, especially in the context of antimicrobial resistance. This classification helps us think strategically about how to combat bacterial threats effectively. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about how the increased consumption of antimicrobial drugs is affecting both humans and animals. As we see more widespread use, the overuse of common antimicrobials by physicians becomes a concern. Often, this choice is influenced by factors like low cost and perceived low toxicity, which can lead to unnecessary prescriptions. Sometimes, antimicrobials are prescribed improperly, such as starting with broad-spectrum drugs when they aren't really needed, or when those drugs turn out to be ineffective against the infection. This kind of prescribing can have serious consequences. The key danger here is that overusing antibiotics in humans promotes the development of resistant organisms. In other words, bacteria can evolve to become resistant to the drugs we rely on. Plus, if a patient has taken antimicrobial drugs before, they're at a higher risk of subsequent infections with drug-resistant organisms. All of these factors, overuse, improper prescribing, and prior exposure, contribute to the growing problem of antimicrobial resistance. It's a complex issue that ties directly into our overall goal of promoting responsible antimicrobial use to safeguard effectiveness for the future. When bacterial cells are exposed to an antimicrobial agent, there are essentially two possible scenarios. The first scenario involves cells that are resistant to the antimicrobial agent. In this case, the non-resistant cells are killed off, leaving only the resistant cells behind. When we then allow these resistant cells to regrow, all of the cells in that culture will be resistant, which highlights how resistance can be passed on through the population. The second scenario involves persister cells, which are a bit different. These cells are not resistant in the traditional sense. They are dormant during treatment, meaning they're not actively growing or dividing. When the antimicrobial agent is applied, the non-persister cells are killed, but the persister cells survive because of their dormant state. Once the environment is favorable again, these persister cells can wake up and start growing. However, it's important to note that they are not resistant. If they are exposed again, they can still be susceptible to the antimicrobial agent unless they develop resistance over time. This distinction between persistence and resistance is crucial because it influences how we approach treatment strategies. Resistance involves genetic changes that make bacteria inherently resistant, while persistence is about a temporary dormancy that allows bacteria to survive treatment. Understanding this helps us grasp why some infections are so difficult to eradicate completely, and why they can recur even after seemingly successful therapy. When we look at bacteria as a group, or even as a species, they're not necessarily uniformly susceptible or resistant to a particular antimicrobial agent. In fact, resistance levels can vary quite a bit even within related bacterial groups. This variation is an important point to keep in mind. Now, when we measure how susceptible or resistant bacteria are, we typically look at something called the minimal inhibitory concentration, or MIC. This is the lowest concentration of a drug that will actually stop the bacteria from growing. Think of the MIC as a range rather than a fixed point. It represents the average MIC for a species across different strains, giving us a sense of how sensitive or resistant that species generally is. And here's a crucial point. If the average MIC for a species falls into the resistant part of that range, then we consider that species to have what's called intrinsic resistance to that drug. So, understanding the MIC and these ranges helps us grasp the overall susceptibility profile of bacteria, which is vital for choosing the right antimicrobial therapy. Natural resistance can be either intrinsic or induced. Intrinsic resistance is always present in the species itself, meaning it's a natural trait. Induced resistance, on the other hand, involves genes that bacteria naturally have but only start to express resistance after exposure to an antibiotic. When we talk about intrinsic resistance, it's important to remember that this trait is shared universally within a bacterial species. It doesn't depend on whether the bacteria has been previously exposed to antibiotics, and it isn't related to horizontal gene transfer, which is the transfer of genes between bacteria. The main mechanisms behind intrinsic resistance usually involve a reduction in permeability of the outer membrane. For gram-negative bacteria, this often means changes in the lipopolysaccharide layer, or LPS, which acts as a barrier. Another common mechanism is the natural activity of efflux pumps. These are proteins that bacteria use to pump out antibiotics, preventing their accumulation inside the cell. Multi-drug efflux pumps are particularly important because they can expel a variety of antibiotics, contributing to resistance. It's also worth noting that these efflux pumps are involved not just in intrinsic resistance, but can also be induced, meaning bacteria can ramp up their activity in response to antibiotic exposure. Overall, understanding these natural resistance mechanisms helps us grasp how bacteria survive in the presence of antibiotics, and why some infections are so challenging to treat. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about how bacteria can acquire resistance genes. They can do this through several main routes: transformation, transposition, and conjugation. All of these are forms of horizontal gene transfer, or HGT, which means genes moving between bacteria in a way that's different from parent to offspring. Additionally, bacteria can develop resistance through mutations in their own chromosomal DNA. Now, the acquisition of resistance can be either temporary or permanent, depending on how the genetic material is integrated or maintained. One of the most common ways bacteria gain outside genetic material is through plasmids, which are small DNA molecules that can carry resistance genes and transfer them between bacteria. Bacteriophage, or virus-mediated transmission, is less common but still a possible route. It's also important to note that some bacteria, like a Cyanobacter species, are naturally competent. This means they have the ability to directly take up genetic material from their environment, increasing their chances of acquiring resistance. Overall, understanding these mechanisms helps us grasp how resistance spreads and persists, which is crucial in tackling antibiotic resistance effectively. Let's talk about resistance mechanisms now. Intrinsic resistance, which is naturally present in bacteria, often involves limiting the uptake of the drug, inactivating the drug itself, or actively pumping it out through efflux systems. On the other hand, acquired resistance, which develops over time, can include modifications to the drug target, inactivation of the drug, or efflux as well. It's important to recognize that the types of mechanisms bacteria use can vary depending on their structure. For example, gram-negative bacteria tend to utilize all four main mechanisms: limiting uptake, inactivating the drug, modifying the target, and efflux. Conversely, gram-positive bacteria are less likely to limit drug uptake because they lack the outer membrane, known as LPS, and they also generally have a reduced capacity for certain efflux mechanisms. Overall, antimicrobial resistance mechanisms fall into four main categories, and understanding these helps us grasp how bacteria evade antibiotics. Recognizing these differences is crucial as we develop strategies to combat resistant infections. To help fight this growing threat, scientists are actively developing new methods and techniques that play a crucial role in both treating infections and preventing the inappropriate use of antibiotics. These innovative therapeutic approaches include a variety of strategies. For example, phage therapies use viruses that specifically target bacteria, while quorum-sensing inhibitors disrupt bacterial communication, making infections easier to control. Immunotherapeutics boost the body's immune response, and predatory bacteria are designed to hunt down and eliminate pathogenic bacteria. We also have antimicrobial adjuvants, which enhance the effectiveness of existing antibiotics, and hemofiltration techniques that physically remove toxins and bacteria from the blood. Nanoantibiotics utilize nanotechnology to deliver drugs more effectively, and microbiota transplantation restores healthy bacteria to the gut to fight infections naturally. Plant-derived antimicrobials harness compounds from nature, and RNA therapy offers targeted genetic approaches. Vaccine development remains a cornerstone for prevention, and probiotics are widely used to promote a healthy balance of gut bacteria. When probiotics are active in the intestine, they produce compounds called postbiotics, metabolites and structural components, that have various therapeutic applications, especially antimicrobial effects. Importantly, these postbiotics do not promote the spread of antibiotic resistance, which makes them a promising avenue in our fight against resistant bacteria. All these innovative methods are vital in our ongoing effort to combat drug resistance and safeguard public health. Currently, there are three main ways we can modulate the microbiota: through prebiotics, probiotics, symbiotics, or postbiotics. Prebiotics are essentially food for the microorganisms in our gut. They not only feed these beneficial microbes, but can also have a positive effect on our overall health. Some common prebiotics we see today include human milk oligosaccharides, lactulose, and inulin derivatives. On the other hand, probiotics are live microorganisms that directly impact our gut microbiome. They work by delivering beneficial bacteria to our gastrointestinal tract. According to the World Health Organization's definition from 2002, probiotics are live microorganisms that, when taken in the right amounts, can have a positive effect on our health. In clinical practice, the most frequently used probiotics include bacteria from the Lactobacillus, Bifidobacterium, and Streptococcus genera, as well as yeast like Saccharomyces. There have been several meta-analyses supporting their effectiveness in treating various conditions, such as acute gastrointestinal infections and inflammatory bowel diseases. However, it's important to recognize that increasingly, individual reports are questioning their safety and effectiveness, especially in high-risk patients. This has led to growing interest in alternative options, like postbiotics, which are considered a surrogate group of preparations. So, understanding these different approaches helps us better appreciate the complex ways we can influence our microbiota to promote health. Let's talk about the concept of postbiotics. It's based on the idea that the beneficial effects of our microbiota are actually mediated by the metabolites they secrete. These are the substances that microorganisms produce through their metabolic activity. Now, while this concept is gaining attention, it's important to note that the exact definition of postbiotics is still under discussion within the scientific community. In essence, postbiotics include any substances that are released by or produced through the activity of bacteria or fungi. These substances have a beneficial effect on the host, whether directly or indirectly. For example, they might influence our immune system or gut health in positive ways. It's also key to understand what postbiotics are not. They are not probiotics themselves, meaning they are not live microorganisms. Nor are they purely prebiotics, which are mainly food for microbes. Instead, postbiotics are all the beneficial substances of bacterial or fungal origin that don't meet the strict definitions of probiotics or prebiotics, but still confer health benefits. So, to sum up, postbiotics are an exciting area because they offer a different approach to harnessing the benefits of microbiota, focusing on these beneficial metabolites rather than live organisms alone. This ties into the broader theme of understanding how our microbiome influences health and how we can leverage that for better outcomes. Let's explore the concept of postbiotics. Essentially, postbiotics are either metabolites or fragments of microorganisms that provide beneficial effects to the host. In other words, they're the bioactive compounds produced by or derived from microbes that can support health. Now, because of their structural heterogeneity, there are many different techniques used to obtain postbiotics. This diversity means we can tailor the extraction methods depending on the specific molecules we're interested in. One common approach involves the lysis of bacterial cells. This process breaks open the cells to release their contents. Lysis can be achieved through various methods, including chemical techniques like enzymatic or solvent extraction, and mechanical methods such as sonication and heat. Once we've released the cellular components, the next step is to isolate and identify the specific molecules of interest. Techniques like extraction, dialysis, and chromatography are essential here. They help us pinpoint and purify compounds such as short-chain fatty acids, which are key postbiotic molecules with recognized health benefits. Overall, these methods highlight the versatility and complexity involved in acquiring postbiotics, which are becoming increasingly important in the field of functional foods and targeted therapies. Postbiotics display a range of pleiotropic properties, meaning they can affect multiple systems and processes in the body. One of their key actions is inducing the differentiation of T regulatory lymphocytes, which play a crucial role in maintaining immune tolerance and preventing overactive immune responses. Additionally, postbiotics stimulate the synthesis of anti-inflammatory cytokines, helping to calm down inflammation. These actions are important because they help restore the delicate balance between the two major arms of the immune system, represented by Th1 and Th2 lymphocytes. An imbalance between Th1 and Th2 cells is a common factor in many immune-related diseases, including atopic disorders like allergies and asthma. So, by restoring this balance, postbiotics could potentially help prevent or manage these conditions. Another interesting point is the antibacterial activity seen with postbiotics. It's believed that this effect is mediated through their influence on the molecular structure of enterocytes, the cells lining our intestines. This influence helps to strengthen and seal the intestinal barrier, which is vital for preventing harmful pathogens from crossing into the bloodstream. There's also a fascinating, statin-like activity associated with postbiotics. While statins are well known for their cholesterol-lowering effects, postbiotics seem to mimic some of these actions, opening exciting possibilities for their future use in metabolic and related diseases. Overall, the mechanisms of action of postbiotics are diverse and promising. They not only modulate immune responses, but also strengthen gut barrier functions and potentially offer therapeutic benefits similar to statins. This multifaceted activity makes them a very promising area of research for a wide range of health conditions. When discussing the therapeutic benefits, it's important to highlight the superiority of postbiotics over probiotics, especially when it comes to safety. The key advantage of postbiotics is that they bypass the issue of acquiring antibiotic resistance genes and virulence factors. These concerns are particularly relevant in vivo, where probiotics might transfer such genes, potentially leading to resistant or harmful strains. Postbiotics eliminate the need for exposure to live microorganisms altogether, which is especially crucial for children. Their immune systems are still developing, and their intestinal barriers are more leaky, making them more vulnerable. So, in terms of safety, postbiotics offer a significant benefit by reducing these risks and providing a safer therapeutic option. This focus on safety aligns with the overall theme of the presentation, ensuring effective and secure approaches to supporting gut health. Now, let's talk about cell-free supernatants, which are really interesting. These are liquids that contain biologically active metabolites secreted by bacteria and yeast into their surrounding environment. Essentially, after microbes grow in a culture, we can collect these supernatants directly. The process involves incubating the microbes, then centrifuging the culture to remove the cells, and finally filtering the mixture to ensure it's sterile. What's fascinating is that supernatants derived from bacteria like lactobacillus and bifidobacterium have shown antibacterial activity, especially in preventing harmful bacteria like enteroinvasive E. coli from invading intestinal cells in vitro. This suggests their potential to protect the gut. The antibacterial effects might be due to competition for receptor sites, preventing the pathogens from adhering, but they could also have a local effect on the intestinal environment, strengthening the cell barrier or influencing protective gene expression. This makes cell-free bacterial supernatants promising candidates as anti-infectious agents, particularly for conditions like diarrhea. It's an exciting area because it opens up new possibilities for therapies that harness these natural bacterial products without the need for live microbes. Now, moving on, let's look at the current classes of postbiotic drugs available today. Exopolysaccharides, or EPSs, are interesting biopolymers produced by microorganisms during their growth. These are released outside the bacterial cell wall, creating a diverse group of substances with different chemical properties. What's fascinating is that EPSs can actually influence our immune system by interacting with cells like dendritic cells and macrophages. They can enhance the proliferation of T and NK lymphocytes, which are crucial for immune responses. For example, an EPS isolated from tofu, which comes from Lactobacillus plantarum, has been shown to induce nitric oxide secretion and boost the phagocytic activity of macrophages in lab models. This same EPS also increases IgA levels in the intestinal mucosa and stimulates lymphocyte proliferation, indicating a strong immune-modulating effect. Additionally, using an EPS from Lactobacillus casei as an adjuvant can improve the efficacy of vaccines, such as the foot-hand-mouth disease vaccine. It's also worth noting that some lactobacillus strains from fermented durian fruit produce EPSs with antimicrobial and antioxidant properties, adding another layer of potential health benefits. So overall, EPSs are a key component of postbiotics with significant immunomodulatory and protective roles, which ties into our broader discussion on how these microbial products can support health and disease prevention. Many components of the bacterial cell wall are immunogenic, meaning they can trigger a specific immune response. One such component is bacterial lipoidochoic acid, or LTA. LTA is primarily found in the cell walls of gram-positive bacteria and has the ability to be spontaneously released into the surrounding environment. When applied topically, LTA can enhance our body's nonspecific defense mechanisms. This leads to the release of anti-infectious peptides, such as human beta-defensin and cathelicidin, which play crucial roles in protecting against pathogens. Interestingly, bacteria from the genera Lactobacillus and Bifidobacteria, which produce significant amounts of LTA, can stimulate the skin's mast cell response. This activation helps defend against certain bacterial and viral infections. All of this suggests that LTA might have promising potential as a treatment or preventative measure for a wide range of skin infections. So, understanding these bacterial cell wall fragments and their immunogenic properties really ties into the broader theme of how our microbiota and bacterial components can support skin health and immune defense. Bacterial lysates, or BLs, are obtained through the chemical or mechanical degradation of bacteria that are commonly found in the environment. These bacteria are both gram-positive and gram-negative types. The interesting part is that their clinical use hinges on the concept of the gut-lung axis. Essentially, this is the functional connection between our immune system in the gut and the respiratory system. What's really fascinating is that studies have shown that when BLs are taken orally in a lyophilized form, they reach the Peyer's patches in the small intestine. These are important immune sites, and once the BLs arrive there, they stimulate dendritic cells, or DCs. This activation then triggers T and B lymphocytes, which are crucial players in our immune defense. From there, these mature lymphocytes migrate to the mucous membranes of the respiratory tract. Once there, they play a role in initially stimulating the innate immune system and promoting the secretion of IgA antibodies. This process helps strengthen our respiratory defenses from the inside out. And it's worth noting that the safety of using BLs has been confirmed through numerous clinical studies. They've been tested in various diseases, including recurrent upper respiratory tract infections in children, demonstrating their potential benefits and safety profile. So, overall, bacterial lysates represent a promising approach to boosting immune health by leveraging the natural connections within our body's immune network. Some postbiotics can exert direct antimicrobial effects by helping to seal the intestinal barrier, which is crucial for preventing harmful substances from entering the bloodstream. They can also bind competitively to receptors that some pathogenic bacteria need to attach and infect the host, effectively blocking their colonization. Additionally, postbiotics can influence the expression of host genes, modulating immune responses and maintaining gut health. They also help modify the local environment within the gut to make it less favorable for pathogens. Research has shown that combining postbiotics with probiotics can be especially effective. For example, in a preclinical model, such a combination successfully prevented rotavirus-associated diarrhea, which is a common cause of severe illness in young children. Moving to clinical evidence, randomized trials with children aged 12 to 48 months demonstrated that daily intake of products containing L-pericase postbiotics led to a significant reduction in various illnesses. These included diarrhea, acute gastroenteritis, and respiratory conditions like pharyngitis, laryngitis, and tracheitis. One of the key components here is butyrate, a short-chain fatty acid which supports the regeneration of the intestinal lining, crucial for restoring gut integrity after infections. We also see that supernatants from probiotic strains like L. rhamnosus, GG, can protect human intestinal smooth muscle cells from damage, highlighting another mechanism by which probiotics support gut health. Lastly, bacterial lysates have been used in children and are associated with a notable reduction in respiratory tract infections compared to controls. This underscores the potential of microbial-derived products, not just for gut health, but also for protecting the respiratory system, tying into our overall theme of infection prevention through microbiota modulation. Let's take a moment to consider the potential role of postbiotics in preventing and treating SARS-CoV-2 infection. Recent research suggests that the structure and metabolic activity of our intestinal microbiome could be linked to how our bodies respond to COVID-19. In other words, the microbiome might influence the presence of biomarkers that predict a severe course of the disease. This highlights an exciting area where modulating the microbiome with postbiotics could potentially help reduce the severity or even prevent COVID-19. It underscores the broader theme of how our gut health is interconnected with immune function and infectious disease outcomes. So, understanding and harnessing these microbial interactions could open new avenues for supportive therapies in managing COVID-19.